Thanks for that introduction. Hello, my name is Garrett Wong, and I'm a manager on the cell biology services team here at Thermo Fisher Scientific. I've been with the company for about 6 years, and my team focuses on assay development and advanced cell models for customer-related projects. Today, I'll be giving a talk on engineering patient-derived tumoroids to enable high-throughput screening workflows for immuno-oncology. Immuno-oncology, also known as cancer immunotherapy, is a quickly growing field that looks at how individuals' immune systems can be used to fight cancer. This field aims to develop therapies that can target cancer cells while leaving the healthy cells untouched. Here are some of the immunotherapies that are currently being leveraged. So you have checkpoint inhibitors that target cell signals that some cancer cells use to repress immune response. There are cancer vaccines where you can sensitize the immune system to better recognize cancer cells. Antibody therapies, which bind and block cancer antigens to inhibit growth and promote immune responses. Then you have CAR cells, which are cells that are that are expressing a modified antigen receptor to enable cells like CAR T and CAR NK cells to recognize and bind antigens. And then lastly, there are tumor-infiltrating lymphocytes, which are immune cells that have already migrated to the tumor microenvironment. And then these can be isolated and expanded ex vivo. And then once you have enough cells, they can be reintroduced to a patient to increase the immune response against the cancer. One key way to validate these types of therapies in vitro is a cell-based killing assay to examine therapy's killing efficiency as well as the specificity against their target. One of the main challenges for immunotherapies is finding a model that accurately reflects the cancer biology and the drug response seen in patients. The most commonly used models are immortalized cell lines and animal models. And each of these have their own limitations, including genetic drift and species-specific responses, which might make these less applicable in the clinical setting. Tumoroids or cancer organoids have become an appealing alternative to both immortalized cell lines and animal models due to their ability to capture donor-specific characteristics and also provide a better representation of the tumor microenvironment. Both of these, these models can be used in a variety of different workflows, from biomarker discovery to validation, as well as precision medicine workflows. This slide is an overarching view of the tumoroid workflow. The first consideration that a scientist would need to make is sourcing the tissue. These can come from a variety of different sources. They can be derived from tissue resections or biopsies if a new tumoroid line is desired or a specific genetic signature is needed for their research. But they can also be sourced from a PDX tissue or obtained through commercially available tumoroid lines. Once the tissue has been identified, these tumoroids can be cultured and banked in suspension or embedded for many passages. This established tumoroid line should be characterized to ensure that tissues is genotypically and phenotypically representative of the original donor tissue samples from passage and also from passage to passage. If the experiment requires it, these models can also be engineered to generate a reporter pool or also knock down targets of interest that are relevant to a immunotherapy or drug screening workflow. Both engineered and unmodified parental tumoroid lines can be used in various assay workflows, from drug screens to functional assays to co-culture systems. Today, I'll be focusing on how we leverage tumoroid models for an immuno-oncology workflow, though. In this case, the tumoroid lines were derived from primary dissociated tumor tissue, specifically colorectal cells. These cells were then expanded in suspension and banked. From there, these were characterized and looked at their mutational profile as well as gene expression, comparing the original tissue to the established tumoroid line as well as the tumoroid line over multiple passages. In this experiment, we also engineered these tumoroid cells to express GFP to become a reporter cell line to assess cell viability. And In this system, we would be co-culturing them with some effector cells, and in this case, we were looking at primary NK cells that were isolated from PBMCs, as well as an NK92 immortalized cell line. Both of these cell lines would be these cells would be expanded, and the primary NK cells were characterized using flow cytometry, both for purity as well as the NK cell characteristics. At this point, you could engineer your NK cells to express a CAR if you are interested in making a CAR NK cell line. But in this case, we opted out of doing this, and we wanted to look at the cytotoxicity of just primary NK cells and NK92s against the tumoroid lines. In this experiment, the tumoroid cell lines were derived from dissociated tumor cells from primary colorectal tumors. And in this process, the cells were allowed to reaggregate over 2 to 7 days. And then once they have clustered, they were embedded into basement membrane extractomes for the initial growth. Once these formed tumoroids, they were transferred to suspension culture, where instead of being embedded, they would now have the basement membrane extract added to the media solution rather than them being encapsulated. For this process, a tumoroid application note will be released for these early steps through Thermo Fisher, where we can provide some guidance on how scientists can best derive their own models. Some commonly used protocols use media systems adapted from healthy organoid media systems, which may lead to overgrowths of normal epithelial cells, making pure tumoroid line establishment more difficult. The OncoPro media system used in this workflow selectively expands EpCAM-positive cancer epithelial cells. And even when we start with a mixed population of cells from a tissue resection, these tumoroid lines can have a greater than 90% EpCAM-positive cells while retaining their donor-specific characteristics, as you've seen in this figure. Tumoroids can be cultured for multiple passages while maintaining genotypic and phenotypic characteristics of their original tumoroid tissue. The figure on the left shows 3 different colorectal lines, so the 1044, 3209s, and 021320. Looking at different the genomic mutations from a targeted DNA sequencing panel, OncoMine. This shows that the patient-specific mutations are consistent between the different passages. As you can see here, you go from the donor tissue at P0 to P50, and these are looking at the single nucleotide variants. There's also a heat map down here representing clinically relevant mutations, also called by the OncoMine software. And you can see that these are mostly conserved over time. There might be some drift seen and sequencing noise that may be observed here, but overall, it looks like these are very consistent over multiple passages. And then on the right, this figure demonstrates the gene expression signature between the tissue. So this is T, denoted by a T, and the organoid or tumoroids denoted by an O. And so for T1 and O1, those are both for the same donor, and then T2 and O2 are the same donor. And in this figure, this shows the 50 most differentially regulated genes, but we still see a strong correlation between these two sets, even though they are the 50 most, the top 50 most differentially regulated, giving us a lot of confidence that these are stable and representative of the donor tissue. After this initial tumoroid derivation, tumoroids are maintained in suspension culture, and they're fed every 2 to 3 days and passaged every 4 to 14 days. For us to classify the cells as a established tumoroid line, we have some stringent criteria that the cells need to meet. First of all, they need to maintain those patient-specific characteristics, and that's why we have a lot of characterization via both genotypic and phenotypic signatures. These cells need to be actively dividing, so they need to be split at least every 14 days for at least 5 passages. These cells also need to have achieved at least 5 cumulative population doublings, and they also need to be able to go through freeze-thaw cycles. For this third one, with the 5 cumulative population doublings, that's within the passage. The media system for culturing and banking these tumoroid models is the newly released Gibco OncoPro tumoroid culture medium, and this provides a streamlined workflow for tumoroid culture where there's fewer reagents that need to be mixed together for a complete media system, as well as the ability for it to support multiple cancer indications, as well as its automation friendly and liquid handling friendly culture conditions. While expanding cells for banking, we closely monitor the morphology as seen in the left figure. You can see there is 2 different colorectal lines here, line 1 and line 2 in this example, and they're across 3 different passages. You can see that across these different passages, the morphology is maintained for these colorectal lines, whereas they're still distinct between the 2 different donors. Also, one of the examples or one of the criteria that we alluded to in the previous slide, the doubling time. So here's a graph of the cumulative population doublings seen in the culture for across many days in culture. And you can see that there are stable growth rates between both the 2 colorectal lines here, so the HUCOs, and the 2 lung lines here as well, the HULU lines. These arrows represent the freeze-thaw time points, showing that these cells can recover and also can be stably expanded after cryopreservation. And then all of these lines were taken out to at least 150 days in culture. While may not be something that we typically do, this is just goes to show that these lines are stable within culture. And then on the right, we also show that there are distinct subpopulations within distinct tumoroid lines. So this is a single cell RNA-seq of one of the colorectal lines that show intratumor heterogeneity, and you have different cell subpopulations here. Some work is being done to do additional single cell RNA-seq on some of the other tumoroid lines so that we can have a more comprehensive data set for these. The best practice is to routinely characterize tumoroid cell lines as you carry these through multiple passages and before assays to make sure that they have not altered their genotypic and phenotypic signatures. So one way to do this is using the Oncomine assay, which looks at 161 of the most common cancer-associated genes. And here is an example of the Oncomine readout looking at 3 different colorectal lines denoted by the 3 different colors, the orange, blue, and light blue. And in this, there are also the different passages. So you have P13 and then P50. So, and then this one's P21 and P50 down here as well for these different tumoroid lines. And as you can see, the heat map shows a high degree of correlation denoted by these red boxes between the passages of the same donor. But then you can see that when you compare them cross donor, there's very low correlation between the different tumoroid lines, at least for the single nucleotide variants. And then on the right, this is an example of a gene expression panel where that was taken from a paper, basically, at least the 38 gene panel that was ran through the gene expression software, where this colorectal cells could be sorted, colorectal cancer cells could be sorted into 5 different molecular subtypes based on their gene expression. And you can see that in between each of these lines, they each have their own distinct molecular subtype that they are expressing. So for example, the 021320s, they have a high level of goblet-like and inflammatory molecular subtype expression, whereas the 3209 has a more transient amplifying molecular subtype. And then also, if you look at these in between passages, you can see that the molecular subtype that they most resemble typically remains the same. There does seem to be some level for the Q code for 1044. There might be a goblet-like subpopulation expanding in this line specifically. So for long-term maintenance of global of these for long-term maintenance of these tumoroid lines, it's good to look at the gene expression patterns. The panel on the left is a principal component analysis demonstrating that the different cancer tissue types will cluster together, but they most closely cluster together with their own donor. So each color represents a different donor. So you have the red, blue, purple over here. These are all different colorectal tumoroid lines. And then they all represent different passages. And as you can see, they all cluster together with its own donor. While there may be some differences in the passages, but they most tightly correlate with each other. Whereas if you look at different cancer tissues, so these are the squamous cell carcinomas for non-small cell lung cancer, and you can see that these ones cluster very differently than the colorectal, as well as the non-small cell lung carcinomas for the large cell ones down here in the right. And then panel on the right, this is another way to look at gene expression over time across multiple passages. This is looking at the passage 13 versus passage 50 of one of the colorectal tumoroid lines. And you can see that for the differentially expressed genes, there's only about 200 or so upregulated and 200 or so downregulated genes. This is overall less than 3% of the total genes looked at for RNA-seq. And for a heterogeneous population, we believe that this is pretty good for a naturally heterogeneous population over 37 passages. Additionally, we looked at the gene ontology mappings, where we looked at the differentially expressed genes across different cell signaling pathways. And this red line denotes anything that's statistically significant, and we only saw that only 3 of the pathways were above that. So we were pretty confident about this data that these tumoroid lines are not significantly changing over multiple passages. Once the donor-specific signatures have been well characterized, one of the tumoroid lines that we selected were the colorectal line. We engineered using a GFP lentivirus to make a GFP reporter cell line. This would allow the GFP intensity to be used as an indicator of the tumoroid cell viability in time-lapse imaging. And here's a quick overview of the lentivirus production workflow. For transduction, these cells were already over 80% GFP positive, but we wanted to put these through selection to make sure we had a more pure population of cells to be used in the cell killing assay. In the figure on the left over here, we have a fluorescent image taken of the tumoroids in a fluorescent microscopy. And then on the middle, we have the flow cytometry quantification of this GFP pool that we had after selection compared to the parental. And as you can see, there's over 99% GFP positive cells, and it's about 2.5 to 3 log shift compared to the parental population. Since these cells were put through selection, we were concerned that there might be a shift in the heterogeneity of the tumoroid line due to the selection pressure maybe altering gene expression or causing a subpopulation to drop out. But luckily, with bulk RNA-seq, we were able to see that there is about a 99% correlation between the wild-type tumoroids before the transduction and the selected resulting GFP engineered tumoroid pools at the end. These cells were left as a cell pool, since any type of clonal isolation would ruin the heterogeneity of the tumoroid line. So typically we would not recommend doing any type of clone selection for tumoroid models for that reason. Now that we have our reporter line, we needed to make sure we had some effector cells that we could use in this immuno-oncology cell-based killing assay. And for those effector cells, PBMCs were isolated from a leukopak for the primary NK cells. And these cells are further isolated from the PBMCs using dynabeads. And then they were expanded using the NK expander media system. And then as a side note, we also had NK92, the immortalized cell line. Those are also being expanded alongside these, but obviously those do not need to go through this type of process where you need to isolate them from a leukopak. Once the NK cells were expanded, the primary NK cells at least were characterized via flow cytometry. And in this figure, the CD45 and CD56 positive cells identify these cells as NK cells as well as lymphocytes. And then since the population is CD3 negative, these cells can be identified as NK cells rather than NKT cells. And then lastly, the CD16 positive positive population are ADCC NKs, whereas the CD16 negative cells are interferon gamma NK cells. Now that the reporter cell pool and the effector cells have been expanded, these cells are ready to be assayed. For the cell-based killing assay, tumoroids were seeded and allowed to aggregate first, and then 72 hours later, the tumoroids were stained for caspase 3.7, which was red in this case, and the NK cells or NK92 cells were added on top of the cells at differing effector to target ratios. And then throughout this entire process from day 4 to day 7, these cells were live imaged to look at the GFP intensity and the caspase activity change over time. And then also at the end of this assay at day 7, the supernatant was also harvested and used for a cytokine profiling assay. Here are some of the results for the killing assay. On the left side, there are images that were taken 24 hours after the addition of the primary NK cells. And you can see that the GFP and the caspase channel here, and then there's the merged channel to the right of that. Each row represents differing effector to target ratios, with the bottom one being the tumoroid cells only with no effector cells, and then it would increase in the amount of effector cells added to each condition. So this is 0.3125 and 0.625 effector to target ratio. And then even looking at the fluorescent images without quantification, you could clearly see that there is a drop in GFP intensity as you increase the effector to target ratio, as well as an increase in the caspase red activity in a dose-dependent manner. And then on the right, this is just a quantification of these time-lapse images that we took. And this has both the primary NK and the NK92 cells quantified for this. On this figure, you can also see that there is you can see the intensity increase over time, because this was actually, we had time-lapse imaging going on before the NK cells were added. So this dotted line represents the time point at which the NK effector cells were added to the tumoroids. And then you can quickly see that the GFP intensity drops off in the primary NK cells after the addition of the immune cells. And then you can also see a peak in the caspase 3.7 red intensity. And you can clearly see that this is in a dose-dependent manner, because with the highest effector to target ratio, which is the dark blue dots, you see the fastest decline in the GFP intensity. And then as you go to lower effector to target ratios, you see the cells die off a lot slower. Another takeaway from this figure is the difference between the primary NK cells and the NK92s, which further demonstrate the need to preferentially work with, or the desire to preferentially work with primary tissues, because the NK92s required both more time and a higher effector to target ratios to reach the same drop in GFP intensity with the primary NK cells. Once the time-lapse imaging was complete, the supernatant was collected to be used in the cytokine release profiling assay. In this case, we use the 65-plex Procardiplex panel. And right here, we have some of the cytokines that are highlighted that are all important for NK-mediated immune response. Typically, you would like to see a dose-dependent increase across the conditions, like you see with the interferon gamma and the trail. But our desire to multiplex this assay caused some complications that we think are happening here, where you see that there is a saturation in signal, like seen in the MIP1 alpha. And for the also for the highest effector to target ratios, we also see some decay in signal, which is not something that you typically would see. So like for IL-3. And based on the previous slide, you could see that most of the GFP signal in the primary NK cell populations, with the highest effector to target cell ratios for the primary NK cells, almost all the GFP signal was gone after 48 hours, indicating all the target cells were killed off. This would mean that many of these cytokines would either stop being produced, and some of these could also degrade over time, as some of these have pretty short half-lives, which could explain some of the discrepancies that we're seeing here, where you have the NK92s have much higher amounts of some of these cytokines. In the future experiment runs, the team will be testing different harvesting time points to make sure that these saturated signals are not occurred in future runs. So what are the key takeaways? We showed that we could establish tumoroid models from patient-derived colorectal adenocarcinoma tumor cells. These cells were also characterized to make sure that they resemble their donor tissue. Then these were engineered into GFP reporter lines. This could be any type of engineering, but in this case it was GFP. And then we were able to demonstrate a proof of concept study on how tumoroids could be used for more relevant target populations and functional assays for immuno-oncology workflows. While in this example, we used primary NK cells as the effector cells, different immune cells can be used. So this could be easily translated to CAR NKs, CAR Ts, or this could also be translated to any other type of immuno-oncology workflow. It just requires some of the parameters to be changed. And so, for example, another potential use case would be knocking down specific targets or overexpressing targets to more easily validate CARs against those tissues, because then you could have otherwise isogenic populations where you have only one target missing, and so you could easily determine your CAR specificity for these tumoroids. And the use of these tumoroids open doors to developing better treatments more quickly, so we could also get a better understanding of what this should look like in the clinic. I would like to thank you for tuning in to this webinar, and now I would like to open up the floor for questions and answers.